The story and research of Elizabeth H. Blackburn, a Nobel Prize winner. Today, I will be talking about Elizabeth H. Blackburn. Elizabeth is a PhD researcher, medicine laureate, and professor that specializes in the study of telomeres and enzyme telomerase. Elizabeth was born in Hobart, in Tasmania, Australia, and raised in Launceston, also in Tasmania, Australia. Elizabeth was born into a family of physicians, and from a young age she was a nature enthusiast, which spiked her interest in the field of biology. In elementary school, Elizabeth discovered her love for playing the piano and strongly believed that she would become a musician. However, as she stated, she recognized that she was competent rather than greatly talented in playing the piano, so she decided to focus on science instead. She studied biochemistry at the University of Melbourne, where she also received her master's degree. In 1975, she received her PhD degree from the University of Cambridge. Later, she decided to follow her husband and went on to Yale University, where she got her postdoctoral degree. Elizabeth discovered the molecular nature of telomeres, which are protective caps at the end of chromosomes that preserve our genetic information. Her main goal as a researcher is to see how telomere and how the enzyme telomerase relate to aging on a cellular level. She studied the effects of stress in mothers and their cell health. She discovered that more stress leads to shortening of telomeres and ultimately leading to premature cell aging. In a research article from 2004, she researched how stress in women causes premature aging. She stated that women experiencing the highest levels of stress, on average, have telomeres that are shorter by the equivalent of at least 10 years of additional aging compared to those with lower stress levels. These findings suggest that at a cellular level, stress may accelerate aging and contribute to the earlier onset of age-related disease. For this experiment, Elizabeth used a group of mothers aged 20 to 50 in the premenopausal stage of life, who have at least had one child. She made sure that women were free of any chronic illness and accounted for their BMI while doing the research. The blood was drawn during the first 7 days of the follicular stage of the menstrual cycle, and morning fasting was necessary for this experiment. The figure was taken from her research article. The top graph shows that women with the highest level of stress have a lower telomerase ratio, while the bottom graph shows that women with high stress levels have lower telomerase activity, further supporting her hypothesis on the connection between telomeres and stress levels. In 2009, Elizabeth received the Nobel Prize in Physiology or Medicine, and in 2012, she received the American Institute of Chemists Gold Medal. These are just two of her many achievements. Currently, she is a professor at the University of California, San Francisco, in the Department of Biochemistry and Biophysics, and is the president of the Salk Institute for Biological Studies, which is a top 5 elite research institute. She is still doing research on telomeres and their impact on aging. I found out about Elizabeth through this project and was amazed by her research, resilience, and character. Like me, she moved to the United States to continue her education and career. She gives hope to cancer patients and shows support to women in high-stress environments through her research. I greatly admire Elizabeth as a female researcher because she continuously proves that women can be equally successful in biological fields. Elizabeth serves as a great example of powerful women in the field of biological research and should be example to the girls and women around the world.